evening and welcome to UCLA's first ever Grad Slam competition. The Grad Slam competition was started at a couple of our sister campuses in the University of California a couple of years ago, and this year the graduate deans at all 10 campuses decided to expand the competition to make it a system-wide one, culminating in a system-wide final round to select the best of the best. So Grad Slam is really a fun way for us to encourage our graduate students to develop a skill that all of us as professionals need to have. The ability to give what's often called the elevator pitch. So that's the short, substantive response you give when you meet someone at a reception or in the lobby of a conference or maybe an elevator, and that person says to you, "Tell me about your work." Now the person asking could be a Nobel Prize winner, could be a venture capitalist, could be a future employer, could be your fiance's grandmother, could be anybody. But we should all be able to explain in a nutshell what we do, why we're passionate about it, and be able to get another person interested, maybe not as excited as we are, but at least a little bit excited about it, and whoever that person might be. So at UCLA, we announced our competition a few months ago, and nearly 60 students committed to competing. Now, this is a skill that's taught a little bit more often in some parts of the campuses than others, so we provided some workshops and some one-on-one coaching sessions, and we encouraged all of our competitors to practice in front of their peers and their friends and maybe their fiance's grandmother. We held 10 preliminary rounds. From each of them, 2 students moved forward to the semifinal rounds. We had 2 semifinal competitions earlier this week, leading to the selection of the 6 finalists you're going to hear from tonight. They represent the top 10% UCLA's best of the best. So I want to thank all of the participants. They're named on the back of your program. Let's give all these brave competitors a round of applause. And I also want to thank the postdocs, the staff, the faculty, the alumni, and the friends who generously gave their time to participate as judges in our earlier rounds and also this evening. So their names are also in the back of your program. Let's give them a round of applause. I also want to especially thank the Grad Student Association because they're our partner in this venture. They've helped us with publicity and many other ways. The president, Mike Hirschman, couldn't be here tonight. Do we have members of the GSA here tonight? A few, maybe? Ooh, they will be. Beer comes later. So I want to thank them for their partnership. And also, I want to thank the staff in the grad division, Assistant Vice Provost Sam Brisola, Sonia Gavin and her team, Latricia Rogers and Alma Zavala, and the many, many other staff who you've seen tonight and behind the scenes who made all the events possible. So let's give them a round of applause. Thank you. All right, so let me say a little bit about how tonight's going to work. First, each of you should have a ballot. If you don't have a ballot in your hand, please raise it and we'll get a ballot to you. A ballot, pencil, or pen will do. Because each of you is also a participant as a judge. Each of our 6 contestants will have 3 minutes to tell you about their work using no more than 3 slides. Each talk will be scored. I sound like I'm reading for Chopped or something. Each will be scored with points for clarity. So did the speaker provide adequate background to help you understand what's important about the work? Was it organized? Did it follow the logical flow? What was their delivery? The pace, the enthusiasm, the body language, their visuals, if they had them. Were they well designed and did they synchronize well with what they're saying? Was it appropriate? Was it free of jargon? And no one's a better judge than you. Okay. Intellectual significance. Did this speaker explain why it matters? And finally, engagement. Did they spark your curiosity and interest in learning more? Okay, so those are the criteria. So after each of the 3-minute talks, we'll give the judges, who are up here, and I'll introduce them in a moment, a few minutes to prepare their score sheet. So we'll do a few-minute break, we'll hand off the mic, you can chill for a few seconds, and then we'll start the next presentation. And after the last presentation, we'll have a short intermission. We'll hang out in this room probably, we can wander around a bit. It might take about 10-15 minutes, you can stretch in room. We'll have some on-screen entertainment also. And then I'll announce the third, second, and first place winners. Then we'll adjourn. We have some refreshments over at the grad bar. There we go. And then we'll go over there. Around 7:15, we'll have the results from you. So we'll collect your ballots as you walk out, and we'll have the audience choice winner. Okay. So our distinguished panel of judges includes, as I introduce you, stand, be recognized, and we'll thank you and welcome you. Don Downey, who's the Director of Development in the UCLA Graduate Division. Thanks, Don. We have Scott Naslin, who's District Director in the Office of California Assemblyman Sebastian Ridley Thomas. We have Lori Policione, who's a partner in Foley & Lardner in Los Angeles and past president of the UCLA Alumni Association. We have Srinivas Pullavarti, who's President and Chief Investment Officer of the UCLA Investment and board member of the UCLA Foundation. And finally, we have Judith Smith, who's Emerita Professor of Integrative Biology and Physiology, Emerita Dean and Vice Provost for Undergraduate Education. Thank you, Judy. Okay. Now, why did these students compete? Aside from glory. All of our finalists will receive a Grad Slam certificate and a $50 gift card for the UCLA store. Now, our distinguished panel of judges will select the top 3 prize winners, and you all will select the audience choice winner. All of those winners will receive a great prize package that includes an opportunity to present in a Grad Slam showcase at the UCLA Career Center Masters and PhD Career Conference Thursday, April 30th at 4 p.m. in the Ackerman Grand Ballroom. An opportunity to present at an upcoming LA Nerd Night, which is coordinated by the Society of Postdoctoral Fellows. And Nerd Night is a monthly event. It's held in more than 50 cities around the world. And presenters give a short, fun, informative talk, presentations in all disciplines, kind of like this, where the audience actually drinks along. So it's like Discovery Channel with beer. And finally, all winners will get dinner with me. So I'm the Dean and Vice Provost of the Graduate Division. Also joining us will be Executive Vice Chancellor Provost Scott Waugh, GSA President Mike Hirschman, and other VIP leadership. Okay. In addition, the Audience Choice Award winner will receive a $500 fellowship. So choose wisely. The third place winner will receive a $1,000 fellowship. The second prize winner will receive a $2,000 fellowship. And the grand prize winner at UCLA will receive a $3,000 fellowship and a paid round-trip travel to compete in the Oakland UC-wide Grad Slam on May 4th, where he or she will compete for additional fellowship prizes. Okay. So that, I think, we are about ready to begin. So first, be sure you have a ballot in your hand. So take notes as you go, but you'll get to vote at the very end. Okay. And so I'm going to prepare to hand off and introduce our first finalist. And he's a fourth-year PhD student. Oscar Campos is a student in molecular biology, and he's going to be telling us about distorted DNA structure in cancer, lessons from yeast. Please join me in welcoming Oscar.
Okay, well, thank you for the introduction. Today I'm going to tell you about the structure of cells, the structure of DNA, and how those structures are disrupted in cancers. So first, this here is a simplified cartoon representation of a cell. And remember, we have trillions of these inside of our bodies. Now what I want you to focus on in my slides is on this structure here. This is the cell nucleus. And inside of it, that's the DNA. Now, in a normal, healthy cell, the nucleus has a nicely shaped, round structure. Whereas in cancer, the shape is very distorted. It's ugly. The nucleus itself has an irregular shape, and the DNA inside is not evenly distributed. Despite all that we know about cancer, we don't really know how this happens. So how can we hope to solve cancer if we're missing a piece of the puzzle? Now, our lab is focused on understanding how it is that cells do this. How do they control the shape and size of their nucleus to prevent it from becoming distorted? To approach this question, we used a comparative approach. We can learn a lot simply by contrasting how it is that different organisms solve similar problems. Now, in our lab, we specifically compared humans to the microorganism yeast. Now, we can make this comparison because although humans and yeast don't look anything alike, at the microscopic level, we're actually quite similar. Not identical, but similar enough that we can make this comparison. Now, when you compare humans and yeast, what you find is that the human DNA is very tightly packed inside the nucleus, and the nucleus is small. Whereas the yeast DNA, it's very loose, and the nucleus is bigger. So we searched for molecular differences between humans and yeast that could explain these structural differences. And we found that the difference could be explained by molecules called histones. We know this because when we change the histones in the lab from the humans to make them look more like yeast and vice versa, we find that the nucleus also changes correspondingly. Now, why is this an important result? Because it turns out that in some cancers, the human histones have changed to look more like the yeast histones. And because normal histones are important for normal structure, altered histones may indeed be responsible for the distorted structures that we see in cancer. So while these findings are an important finding for understanding how it is that distortions occur, we're still stuck with the answer of, are these distortions actually important? Do they contribute to the cancer phenotype? And so by answering those questions, we hope to fill in those missing pieces of the puzzle and hopefully get better opportunities for treating the cancer when it occurs. Thank you.
So I'm pleased to introduce to you fifth-year PhD student Sarah Hirschman. She's in the Neuroscience Interdepartmental Program and Psychology, and her talk is about using lasers to eliminate acetylcholine's role in fear memory. Sarah.
Think back on your strongest, most intense memories. Odds are good that memory is either happy, sad, or terrifying. I study fear memory for two reasons. The first is that fear memories have to be strong to be useful. If a mouse doesn't learn to fear a cat after the first experience, that mouse isn't going to survive long. Secondly, excessive fear in humans can lead to disorders such as PTSD. After the events of 9/11, over 10,000 firefighters, police officers, and civilians were diagnosed with PTSD. Many of these individuals still suffer symptoms today, ranging from difficulty sleeping to inability to return to the site of the event, to terror at the idea of flying in an airplane. But in order to reduce the fear for these individuals, we have to understand the neural mechanisms behind fear learning in the first place. I study the hippocampus, which is a region in the brain that's involved in spatial navigation and forming new memories, which makes it an ideal target for reducing fear. The hippocampus uses many neurotransmitters to communicate, but my focus is on acetylcholine. So acetylcholine increases when we explore a new environment and decreases when we recall old memories. So I thought, what if acetylcholine is the record button on our memory? And if by increasing it, we can make a stronger memory? So I set out to test this idea with a little help from my subjects. So I had some mice explore a new environment. The smell, the feel of the good floor, basically learning about that environment. In some of those mice, I increased acetylcholine in the hippocampus using lasers. Now the following day, those same mice returned to the same environment, and very quickly afterwards they receive a mild foot shock. Now normally that period of time is so short that the mice cannot form a fear memory. So the following day the mice returned to the same environment, and lo and behold, only mice that had acetylcholine increased in the brain actually remember that this is a scary place. So it might seem strange to think of increasing the strength of a memory in order to decrease fear. But actually one of the challenges with human therapeutics for PTSD is that the memory that they form in therapy is not strong enough to compete with fear memory. So one thing that can be done is we can use drugs that increase acetylcholine, administer those during therapy, and perhaps we can make that safe memory be strong enough to compete with the fear memory in order to reduce fear for those individuals. I'm excited to study acetylcholine because I hope that one day we can control our memories, and so that our memories don't control us.
So I'm pleased to introduce our third competitor. He is Brian Odegaard, a sixth-year PhD student in psychology, who's going to tell us about recalibrating your brain, how space and time influence the world inside your head.
In every moment of your waking life, your brain does a spectacular job of binding your senses together to give you a coherent picture of the world. For example, if you're on a hike in Topanga Canyon, your eyes can show you the bright blue sky, your ears can reveal the grass rustling in the wind, your nose can tell you what flowers are in bloom, and, depending on how intimate you'd like to get with nature, other senses can also provide information about the surrounding world. Your brain binds all of your senses together to show you this multisensory world as one coherent image. Now, as a scientist, I'm interested in one specific question. Can we change the brain's tendency to bind? We know that in typical brains, different senses are processed in different regions of the brain, and in most cases, these regions communicate well to piece it all together. But in certain psychological disorders, like autism and schizophrenia, we see abnormalities in sensory binding. So we need basic science to tell us what the rules are the brain uses to bind and how they can be updated. To explore this experimentally, I simplify the complex sensory world around you in the following way. I sit people in front of a large black piece of cloth, and I ask them to localize briefly presented flashes of light and bursts of sound. Flashes of light are displayed on the screen by a projector, bursts of sound come from speakers behind the screen, and people scroll with the mouse to tell me where they think things happen. Now, if lights and sounds are played from close together, people will often tell me these things come from one location. But as I play them from farther and farther apart, eventually this breaks down and people will tell me they come from two locations. So using this paradigm, I tried to come up with ways to increase the range over which the brain would bind lights and sounds. In my first experiment, after asking people to localize things to get an idea of the range over which they would bind, I then played lights and sounds from the same time and same place over and over and over again for 10 minutes to teach their brains an association between these two things. When I tested their localizations again, I found that I hadn't changed the tendency to bind, so I went back to the drawing board to try something else. In a second experiment, again after asking people to localize things, I now played sounds and lights at the same time but at different places over and over and over again for just 10 minutes. This, surprisingly, increased their tendency to bind, meaning that now, even when I played lights and sounds from separate locations, people would tell me they were coming from the same place. In a final experiment, I played sounds and lights at different times and different places, and this did not change the tendency to bind. So what we learn after all of this is that to teach the brain to bind more, give it pieces of sensory information it would usually segregate, but teach it a relationship between these two things, that they happen at the same point in time. In psychological disorders where the ability to bind starts to break down, it's these types of experiences that can teach the brain how to bring it all back together. Thanks for listening.
I'm delighted to introduce our fourth speaker. He is a second-year master's student in electrical engineering. Please welcome Jean-Paul Santos, who's going to talk about how to talk to Mars.
When I was a child, I was fascinated by space exploration. From Neil Armstrong's first walk to the moon to the Columbia space missions, I was captivated by how we understood the origins of our universe. These missions are often supported by new technologies we develop every day, such as robots like the NASA Mars rover. These robots are often sent to other planets like Mars to investigate possible signs of life. Now, just as we use our cell phones to communicate with our friends and family for faraway distances, NASA scientists use antennas on the Mars rover to talk with the rover so that the rover can send images and video back to Earth. Now, the antennas on the Mars rover right now are not strong enough to send the data directly to Earth. What happens is that this NASA rover first calls this large satellite antenna, who then sends the data back to Earth. Now, this way of sending the information back to Earth is reliable. But the only problem is that this large satellite antenna is very expensive to maintain. That is why the goal of my research is to develop a small but powerful antenna that allows the Mars rover to directly communicate with Earth, saving NASA millions of dollars. Now, this antenna design is actually very complicated because antennas that can typically communicate for faraway distances are physically very large. Now, physics tells us that the larger the antenna, the more power it can handle. More power, more reliable calls. Unfortunately, for a Mars rover, large satellite antennas are very bulky and quite enormous. Adding to this complexity is the need for this antenna to be continuously pointed to that of Earth's antennas at any given moment in time. Such an antenna that has all of these traits will help solve a puzzling paradox that antenna engineers have been trying to solve for decades. Small, but powerful antennas. In my research, I was able to develop an antenna that has all of these traits. My antenna is actually made up of small antennas. When you group them together, meet the required power levels. This reduces the likelihood of drop calls, and it enables faster download speeds. This antenna is also quite lightweight, 2.9 ounces, and 10 centimeter wide and 10 centimeter long. Also, the novel shape of these individual elements helps do away with complex alignment issues with those of Earth's antennas, guaranteeing reliable calls from Mars. With such a revolutionary design, not only will NASA save millions of dollars, but NASA scientists will no longer have to ask the Mars rover, "Can you hear me now?" Good. Thank you.
Got to go outside to introduce our next speaker, who is a fourth-year doctoral student in epidemiology. Eolyn Wong is going to tell us about opening the black box, pathways from night shift to heart disease.
In our 24/7 world, people work at all hours to make our world running. But sadly, research suggested that compared to people working 9 to 5, people who work night shifts are more likely to get heart disease. I'm not talking about the type of night work that we graduate students do from time to time, like pulling all-nighters to finish a project due tomorrow. These people could be working night shifts for years because our society needs them. They are there to provide us products, services, and keep us safe. Are these people doomed then? How can we help? Traditional analysis focused on counting how many of these healthy people end up having heart disease, but how they end up here remains in this black box that we don't know much about. My research aims at opening this black box. How do I do that? First, by consulting the literature, I identify some key factors that lie in between night shift work and heart disease. Based on their interrelations, I can connect them and draw this roadmap showing how people can go from being healthy to being sick. Then, guided by this map, I can do statistical analysis to analyze the population level data. My goal is to figure out how many of these people end up having heart disease via each path. Moreover, I'm also looking at I'm trying to figure out all these sentinel these factors can be seen as sentinel nodes, and I'm trying to figure out if I were to change the shape of this intervention, how many people of heart disease will I decrease? So now you may wonder, how can this be useful? Knowing this roadmap can open up opportunities to change people's lives. If, for example, that most of the people go through a certain point of being weight gain or hypertension before they reach heart disease, then we may be able to design programs to help them lose weight or maintain their blood pressure, so we can stop them there. If, for example, what's more important is we can introduce vending machines that provide healthy food, so people don't need to go to McDonald's at night, and we keep them healthy in the first place. So eliminating night shift work is mission impossible in our 24-hour society, but it is possible that we block its harmful effect. It's my hope that my research not only have shed light on the opening the black box, but also helps initiate a change in the workplace, so that these people who keep our society running can themselves live a long and fulfilled life. Thank you.
Okay, so I'm now ready to introduce our final speaker. And he is Yibo Zhang from electrical engineering. He's a second-year doctoral student. He's going to tell us about lens-free on-chip microscopes for cancer detection.
When we talk about microscopes, we think of something like this, don't we? For 3.5 centuries, doctors have used these devices to diagnose diseases. But unfortunately, These devices cost easily thousands of dollars and weigh over 30 pounds, which makes it almost impossible for doctors and pathologists to carry them and use them at the point of care, especially for low-resource settings, such as developing countries or remote villages. Why can't we make microscopes smaller and cheaper? Well, as we know, microscopes use lenses to form images, and these lenses can be very expensive. In addition, the lenses have to be aligned according to the rules of physics, which tell us we cannot make microscopes arbitrarily small. So we found a way to eliminate the use of lenses in microscopes to achieve something called lens-free imaging. These lens-free microscopes are quite small. I bet they fit your pocket better than some cell phones nowadays. They cost $20 each and weigh 100 grams, which is just perfect for global health applications. These lens-free microscope setup is based on something called holography. It can be extremely simple. It has a LED light source on the top and an image sensor chip at the bottom, the same image sensor chip that is used in our iPhone cameras. We put the sample directly on top of the image sensor through this slot on the side and capture its hologram. And then we send this hologram to the computer, and the computer software will do the magic to have high-fidelity reconstruction of the object. Using this technology, we have recently applied it to cancer detection, which is a true milestone for us, considering its potential impact and usefulness. On the left is a full-field reconstruction of a breast cancer tissue section. The field of view, or the area that you can look at once, is much larger than a regular microscope. This is so important to pathology applications because those pathologists usually look at large area pathology slides to diagnose diseases. On the right are zoom-in images taken by the lens-free microscope and the conventional microscope. As you can see, they agree so well with each other. And the pathologists look at these images and make essentially the same diagnosis based upon two of them. We are constantly putting our effort into making this technology better and better. And we believe that in the near future, this microscope, this low-cost, field-portable, and high-throughput microscope can help the medical personnel and patients in low-resource settings for global health applications. Thank you.
In no particular order except that's the order they're in the stack. Please welcome back Oscar Campos. Stay up here if you will. Thank you. And there's your stuff. All right. And Sarah Hirschman. Congratulations and thank you. I think they're alphabetical again. Brian Odegaard. And Jean Paul Santos. And Aylin Wong. And finally, Bo Zhang. So, the winner of that outstanding prize package and a $1,000 fellowship is third place winner, Sarah Hirschman. Exciting. The winner of the outstanding prize package and a $2,000 fellowship. Please join me in congratulating Yibo Zhang. Congratulations. And the first winner of UCLA Grad Slam. Please join me in congratulating Jean Paul Santos. Bravo, everyone. Are we ready? We'll hold up for pictures.
Can you tell us why you decided to participate in Grad Slam?
Honestly, just for practicing my presentation and research. You know, because I was looking at the email that my graduate advisor sent to me over, I think it was in March, and I looked at it as, wow, this is a good way to condense my research. That's really complicated. It's about Mars, about talking with other planets, and condensing it to something that the general audience could understand. And I thought many engineers needed that practice, so I thought, why not enter this Grad Slam and take it serious? And lo and behold, I won, so I'm really, really excited about this opportunity.
Have you done any other public speaking?
You know, that's funny you asked that because in high school I did competitive speech and debate. And in high school I actually was the national qualifier for my Nevada district in oratory, which is a similar type of speaking engagement.
So with regard to this competition, did you participate in any of the training workshops that were provided?
Yes, I actually participated in the one where we were in a group setting and the speaker was talking to us about what to do and what not to do. And I think one of the most important things I really got out of that is the where we were put into a six member and then just one person critiquing us about our jargon. My first practice was I included all the antenna jargon. And I just thought, well, maybe just why not? But that's when I learned, wow, even with this type of semi-jargon, that was not good enough. So I had to really struggle with all the vocabulary I need to explain my antennas in such a way that the general public will understand.
Right. So can you tell us, do you have any advice or any encouragement for other graduate students who might think about entering the competition next year?
Yeah, honestly, just have fun. It was such a fun event. Honestly, looking back on all the rounds, it was so much fun. But I think you have to think outside the box in how to explain your research because sometimes graduate students get so involved in trying to complicate and include as much details as possible. But sometimes what I really thought about is, will my younger sister, who's only 13, understand what my research is talking about? And that's what we need to keep in mind as graduate students is, you know, you might be the most smart person out there, but if you can't explain it, you can't sell the idea. So that's very important.
Okay, so as UCLA's first Grad Slam winner, first place prize, how do you feel right now?
I feel really ecstatic. I'm excited to represent UCLA. I'm sure we're going to beat Berkeley and those guys. But I'm just so excited. This is such a great opportunity. I'm just glad that we did this for our first annual Grad Slam. And I'm just looking forward to the competition in Oakland.
Great. Thank you.